our guest speaker for this evening, which is Dr. Jarrod Bailey. Jarrod is the, let me get this right, the Director of Science at a really fantastic organization called Animal Free Research UK. They used to be called the Dr. Hadwin Trust, and they are promoting science, medical experimentation that doesn't involve animals. And I'm sure he's going to explain more in a few moments' time. The presentation, the talk is entitled How, Why Animal Research is Poor Science and What Animal Free Research UK are Doing to Change Things. So without further ado, let me hand over to Jarrod.
Thank you very much, Brian. And thank you everyone for dialing in this evening. I hope you'll find what I have to say very interesting, and I look forward to the questions and discussion afterwards. The first thing I'd like to do is not to alarm you, but is to say that there's an awful lot of slides this evening. I'm going to talk for about 40 minutes, but the reason I've included so much information is that what I really want to do is convey to you how much evidence there is underpinning this, this desire for a shift away from animal research and towards human-specific, human-relevant research. There's a huge amount of evidence. We really should have made much more progress than we have, and I want to try and convey all of that to you this evening. But don't be overwhelmed. The slides come from literally dozens of talks that I've given over the years, hence the number. But what I'd like you to do is just take away the salient points from each of them and just a gist of what I'm saying, and we can delve into some more details at the end. Okay, first a little bit about me. I think I always like to hear a little bit more about the speaker when I attend a talk. My degree and my PhD were both in genetics and molecular biology at my hometown Newcastle University. I then followed that for 7 years as a senior postdoctoral research associate, also at Newcastle. I'm a bit of a typical Geordian, find it hard to get away from the Northeast. I spent 7 years studying premature labour. Why do women have premature babies? Is there a genetic reason for this? Can we work out a mechanism? Is there something we can ever do about this? And that's really important research because it affects their lives. It has lifelong consequences for millions of people every year. I'd always been morally opposed to animal research, and indeed I had turned down some positions over the years because I couldn't be guaranteed that I wouldn't have to do some form of animal research. But I became increasingly interested in the science and the human relevance of animal research, or the lack of human relevance of animal research, and ultimately so convinced that it was such shoddy science that I really wanted to help do something about it. And so for the last 17 to 18 years or so, revealing my age a little bit, I've worked for various groups and organizations, both in North America and in the UK, investigating this very issue. So hopefully I'll be able to move on. Yeah, a little bit as well about who I work for. I joined Animal Free Research UK as the science director just in July last year. So it's coming up a year I've been there. But the organisation has been in existence for more than 50 years. And as Brian said, until a few years ago, it was known as the Dr. Hadwin Trust for Humane Research. And over that time, this is a bit of a busy slide, but I'll highlight the main take-home points. Over that time, we funded more than £10 million in research grants for animal-free research projects. This has been to more than 260 projects that have taken place all around the UK on many, many different topics. And for example, that included £750,000 just in 2020, lots of it to early career scientists, which we think is very important to get early career young scientists into this type of research. And I'll talk a little bit later about what we do as well as funding research. We engage in educational programs and public affairs work and so on, all to try and expedite the huge cultural shift that we believe is sorely needed away from animal use and towards human research in science. These slides really, they're not to show off. These are my publications. It's just really to illustrate the amount of work that I've done, and others have done much work too, the amount of work that has needed to have been done to really prove our point. We're fighting a very big animal research culture worldwide, not just in the UK, and we have to underpin our argument with evidence, and that's what I've been busy doing for much of the last 20 years. So I'd like to say that if you are uneasy about animal research, or if you outright oppose it, you're in very good company. It's far from a niche opinion, and indeed it's growing over time. Many polls show this, and here are two examples. More than 80% of the EU public oppose monkey experiments that cause pain or suffering, and just 16% of the British public accept monkey experiments, even if, for the sake of argument, they result in human benefit. An EU petition to quote-unquote ban vivisection a few years ago got more than 1 million supporters. And all of this public opinion is actually beginning to translate to action among governments. The EU Parliament wants to phase out monkey research, even in the face of vehement opposition from those who do it. I was at a meeting at the end of 2019 where the Dutch government were committed to ending animal research as soon as they thought it was possible. The United States Environmental Protection Agency and Food and Drug Administration and others are publishing roadmaps to move away from animal experiments. And even within science, record numbers of scientific papers are being published that are criticizing animal-based approaches in science, advocating superior non-animal research methods, and discussing ways to transition away from animal use and towards those superior methods. And a huge number of international conferences are either taking place on this very theme or discussing this issue. And huge tomes, some smaller books, some huge tomes are being published, replete with evidence supporting all of this. These are 4 recent books with dozens of authors' contributions, including mine, that have been published in recent years, showing why animal research is bad research and why we need to do something different. And actually, I can, just back to the polls, I can update these with ones we commissioned just in the early part of last year. So a YouGov poll commissioned by Animal Free Research UK found that 68% of respondents in the UK would support a policy ending experiments in medical research in the UK and replacing them with non-animal alternatives. And 70% of respondents would support animal experiments in medical research being phased out by 2040. Now, people are also concerned about animal research because of the scale of animal research. I'll quickly whiz through some figures to convey this to you. It's been estimated that about 200 million animals are used in science every year worldwide. The UK has consistently been in the top 10 animal users in the world. I think at the minute it's the 7th biggest animal user. In Europe, the UK has traditionally been second only to Germany, but the UK has regularly, if not always, used the greatest number of monkeys and dogs, which are animals that raise the biggest amount of concern amongst the public. The trend in the UK of animal use has been up for 20, even 30 years, and this is largely due to the increased use of genetically modified or GM animals. I'll talk a little bit more about these in a minute. Currently in the UK, just under 3 million animals a year are used in science, and half of these experiments are for the creation and the breeding of genetically modified animals. They are solely responsible for the increase in animal use. And another important point, we often hear from those who want to defend animal research that animals are only used when there is no available alternative. Well, there are many reasons to show that this is not true. One of them is that across Europe, 750,000 animals are used every year, not just for which there are scientifically valid alternatives, but for which there are scientifically validated alternatives, methods that have been through a rigorous validation process, and still 750,000 animals are used in the face of these alternatives. So because genetically modified animals are solely responsible for the increase in animal use, certainly in the UK, we must ask, are they actually any better than non-GM animals in science? And I think the answer is a very firm no. These are just some of the areas of research in which papers have been published in recent years showing that GM animals have failed to translate to human biology and or to reflect the human situation being researched. And there are some really big examples here. Neurodegenerative diseases, Alzheimer's disease, Parkinson's disease, cystic fibrosis, muscular dystrophy, MS, diabetes, and many, many more. Despite this, instead of scientists moving towards human-specific research, they've instead put their weight behind huge efforts to make GM animals less bad. And I'd like to talk about this for a little while. You may have read about a technique called CRISPR in the news. And this is the least bad method that's available. I think that's the most accurate way to put it. But the problem is it still suffers from major issues. One is the appalling level of inefficiency. Typically just a few % of animals resulting from these techniques will be born with the desired genetic modification. The others die naturally or are killed because they're not what the scientists want to see. And there's also still a huge issue, and this is the kind of dartboard, if you like, of off-target effects. So one analogy would be going to play darts with a handful of dozens of darts and throwing them all at the board at the same time. You might hit the part of the dartboard that you want to hit, but then you'd ignore all of the other darts hitting all of the other parts of the dartboard and everywhere else that you didn't want to hit. The problem for both for science and for animal welfare is that these off-target effects are numerous. There may be hundreds of them in any one successfully created GM animal. They are known to frequently be cancer-causing amongst other unwanted issues, and it's thought that they actually can't be eliminated from the process at all. So the problem is, if and when this is done to humans, and this is one of the intentions, a frequency of off-target effects of just 0.1% would be clinically catastrophic. The problem is that this technique doesn't just suffer from off-target effects, it suffers from unwanted on-target effects too. So to go back to the darts analogy, it's like playing darts with a bazooka. You might hit the part of the dartboard you want to hit again, but you're also going to take out the entire board, the surroundings, the wall that the dartboard's mounted on, and most of next door. You're going to cause a huge amount of damage. And again, this is a huge problem. The on-target effects are numerous and often undetected. There are, again, pathogenic consequences for the animals, including cancers. And I'm just going to ignore this little last bit just for the sake of time, because it's quite tricky to explain. The next slide is to really reflect this really poor inefficiency and the consequences of all the unwanted effects. How many GM animals suffer and how much? Well, it's acknowledged that at least half of GM animals suffer some degree of pain and or death, some of which can be serious. And I'm going to give you some examples here. These are taken from a GM mouse database, and you can see some pretty nasty things in here. Skeletal abnormalities, tumor growth and metastases, degenerative joints, deafness, blindness, tremors and involuntary movements, brain hemorrhages, internal bleeding, and much, much more. The point is, these animals are bred to suffer both directly and indirectly, and they do suffer. It is a huge issue. Now I'll just give you a little warning for the next two slides. There's some pictures on here that some of you might not want to see of monkeys in labs. I will tell you when you can look back if you want to look away. I think it's important to keep on reflecting while I'm talking about the science, about the ethical argument about the suffering of the animals in the labs. It is very important. So I'm going to show them now. These photos are from a couple of recent investigations by Cruelty Free International, which is a group that I worked for a number of years, which show what really goes on in its monkey experiments in neuroscience. You can see here monkeys with their skulls removed so that electrodes can be inserted into their brains, put in restraint chairs in which they can sit for many hours a day, many days on end, to be experimented upon. They also undergo fluid and food restrictions so that they will cooperate and do things that are part of the experiment for the sake of a desperate reward of a sip of juice or a snack. And these things are known to give some monkeys symptoms similar to human PTSD. And they also uncovered some evidence of withholding some examples of animals suffering from the authorities and contravening animal care guidelines. And just in this slide too, you can see a row of monkeys restrained by their legs, their arms, and by their heads, all within sight of one another. And on the right-hand side, you can see a typical method of getting a monkey out of its cage to get it into a restraint chair, which, not to put too fine a point on it, involves dragging them out by their neck. So it goes without saying, there are huge welfare issues involved in this type of research. If you looked away, you can now look back. So this is obviously a huge ethical concern, and probably the major reason why many people oppose animal research, and especially monkey research. Now, a few years ago, I published a review of non-human primate neuroscience research, which concluded that, based on evidence in almost 200 referenced papers, there was a considerable exaggeration of the contribution that monkey research had made to human neuroscience. Again, huge overstatement of the general human relevance of non-human primate research in neuroscience, and also exaggeration of how basic science results ended up translating to human benefits. At the very same time, there was a huge understatement and downplaying of the contribution of human-based research to neuroscience progress. There was also minimizing of the consequences of species differences between monkeys and humans and how that affected applying monkey data to human biology. And there was also a real understatement of the degree of suffering for the monkeys used in that kind of research. The problem is, when you put all of this together, is that it affects from both ends the harm-benefit analysis. Now, the harm-benefit analysis is something that has to be done before any animal research is approved. And obviously, there's a real issue if you are understating the harms and exaggerating the benefits. And the argument goes, and there's some evidence supporting this that's published, if harm-benefit analyses were done properly, a huge proportion of animal research would never be approved. Now, in a review in 2011 commissioned by funders of biomedical research, the authors agreed actually with much of what we said in the paper, even though it was conducted and influenced by those with an interest in monkey research. They stated, for example, that in neuroscience, there was little direct evidence available of actual medical benefit in the forms of new clinical practice or new treatments. And this might be because no link with medicine actually existed. In other cases, the panel had to agree that it would have been possible to do the research that was done in monkeys in humans. They also said that in vision research, in which many monkeys are used, the actual and potential medical impacts of most of the studies was low. And they had to say that further reviews based on these conclusions of the outcomes, benefits, and impacts of non-human primate research should be carried out periodically. And this really hasn't been done. So our paper, and I'm going to whiz through these are slides from an original longer presentation on this specific topic, but it made the case that powerful techniques are available to keep neuroscience research, an area in which many monkeys are used and which has severe consequences for the animals, to keep it in a human perspective. Many techniques and approaches such as scanning and imaging machines and other non-invasive ways of tracking brain activity are immensely productive and, crucially, not affected by species differences that we know exist between humans and monkeys. And they're also, these techniques are used in many of the areas in which monkeys are heavily used. Here are some more techniques: TMS, or transcranial magnetic stimulation, where a kind of electromagnetic wand is used to affect the brain through the scalp. Electrocorticography, just whizzing through for speed. And the point is this. One of the claims made by monkey researchers that you cannot get sufficient resolution using these techniques in human research, so you must use monkeys to study down to the level of a single nerve cell in the brain. This is one of the claims they made. Now, I know neuroscientists and surgeons who disagree, who believe that the methods, some of which I put on the slides here, available for human research are sufficiently powerful. And secondly, and this is a really important point, single neuron studies are done in humans. Many hundreds, if not thousands, have been conducted by brave research volunteers who are undergoing brain surgery. They elect to have these small arrays of electrodes implanted into their brain for a while so that recordings can be done and research can be done, all with a human focus. Now, as I've said, we know we must keep a human focus because it's unreliable to apply monkey data to humans, and many and increasing species differences in gene expression between humans and monkeys are known that affect the function of neurons and the brain as a whole. And just briefly, here are some examples. So one study looked at thousands of genes in a part of the brain called the cerebellum and found that 15% of those genes were expressed differently, and this really, really matters when it comes to nerve and brain function. You don't need to know what they are, but molecules called microRNAs, these are little molecules in all of the cells of our body that act as genetic switches on our DNA, so they turn genes on and off and turn their activity up or down. There are hundreds of them, and 1/3 of them diverge significantly between the human brain and the macaque brain, and dozens of them are present in one species but not the other. Again, this really, really matters. And again, you don't need to know what AI editing is. It's an integral part of gene expression, but this is the point. This happens at a higher rate in humans compared to monkeys, especially in the brain, and it is known to affect neuronal functions and neurological diseases. We have to maintain a human focus. We have to study human biology. I'm going to move on now for the sake of time to something completely different, and this is animal testing of new drugs, which, as I'm sure you know, is ostensibly done to protect human safety. Now, there is still no evidence, I would argue, to show that this has any scientific predictive basis whatsoever. Back in 2009, we decided, my colleagues and I, to do our own study using publicly available data. And this is important because a lot of drug study data is in the vaults of pharmaceutical companies. Very little of it is published. We managed to get our hands on data for more than 2,300 drugs for which there were data in humans and in the animals those drugs had been tested on. And we showed that, among other things, for example, if you test a new drug on a monkey and that test suggests that drug will be non-toxic, or in other words, safe in humans, that test adds almost no evidential weight, no weight of evidence to the existing probability based on other tests and data that that drug will be safe in human beings. So we can quantify this. I'll show you here. Let's say other tests and other data similarity to other drugs suggest that a new drug will be 70% likely to be non-toxic or safe in people. If you test that in a monkey and you get the result you want, this drug does not look to be toxic in the monkey, that raises the probability of human safety from 70% to just 70.4%. It's negligible. That monkey test is not worth doing. We found the same in dogs. Again, same drug, 70% likelihood of safety in humans. A negative and encouraging test suggesting no toxicity in dogs. It raises the probability of human safety from 70% to just 72%. Negligible. It's not worth doing that study, which is, of course, a great financial cost and a great cost to the dogs and the monkeys involved. Fortunately, there is now increasing pressure from many sides for the pharmaceutical industry and for the regulators to change this, I'm pleased to say, and to move away from unreliable and downright dangerous at times animal tests. So there is some encouraging movement there. Now, we initially set out to do this on dogs. This was a project funded by ring-fenced funds for dog research. So as part of our campaign against dog experiments, we decided to look at the science that underpins the ethical argument why we shouldn't be using dogs in invasive research experiments. So a few years ago, I authored a review of some really elegant research done in the United States where people's companion dogs are taken into a lab with an fMRI machine. You can see it there. They're trained to sit still in the MRI machine just for a little while where their brains are being imaged. And the neuroscientists involved have shown really, really advanced cognition and emotion in dogs. Now, of course, this should be obvious anyway to anybody who's ever had a dog or who's ever known a dog, but of course this hasn't stopped their use in experiments. And these are some of the things they found, which really is remarkable. Dogs don't just know what we say by the words we say. They can distinguish between tones of voice and recognize emotions in speech. Now, this is cognitively really advanced. It was only known in primates previously. They can also really sensitively interpret facial expressions. And again, this is advanced. Not only that, they can put these two things together to really work out who somebody is, where somebody's coming from, what their mood is, and what their intentions are. This is really advanced stuff. They also showed that dogs really, really experience positive emotions, and they do so much in the way that we do. They have the same brain firing patterns in the MRI machine. Dogs feel empathy, sense sadness and fear, and demonstrate very deep, genuine human bonding. This is all, so we know this anyway, let's face it, but it's been proven with these beautiful experiments. Now, this has led the neuroscientists to say that dogs perceive the world much in the way that we do, which I think should make anyone's blood run cold when you think of dogs in laboratories being experimented upon. And it also led one of the scientists involved to say that dogs have the same level of understanding and awareness as a 3-year-old child. So all of this really reinforces the scientific argument that we're not getting any useful data from dog experiments, that this is the ethical argument that we shouldn't be doing those experiments anyway, and you put them together, and I think you have a very, very formidable argument that we're trying to use to change things. Quickly, I'm probably running over time. I'm going to try and whiz through these things as quickly as possible. Again, it's just to show you the breadth of the evidence. We've been looking for an AIDS vaccine for decades, and we still don't have one. Even though this analysis that I did in 2008 and recently updated showed that at the time in 2008, there were nearly 100 different, often very different, very diverse types of AIDS vaccines that had been developed and tested. They had all looked really promising and encouraging in animals. In nearly 200 clinical trials, none of them worked in people, none of them. And when I updated this a couple of years ago, there'd been about 800 clinical trials of even more HIV/AIDS vaccines, and we still don't have one. We need to do something different. The animal testing isn't working. Alzheimer's disease is also floundering due to too much focus on animals. Animals don't get Alzheimer's disease. And indeed, one of the mainstays of Alzheimer's disease research, the so-called amyloid hypothesis, heavily based on animal research, is now being widely questioned with respect to its human relevance, amid calls to consider moving away from it. And again, there are good reasons for this. We know there are different neuron types involved in humans that many animal species don't have. We know there are different genes and gene expression involved in animal and human brains. And this is what has led to multiple drug and vaccine failures for this disease. Another is stroke, another huge failure. More than 1,300 potential interventions tested on animals, looking promising, and of the few hundred tested in humans, nothing works. We need to do something different. Another issue, which is Parkinson's disease, in which the animal models, again, are also being questioned. Now, to make some of the most used animal models used in Parkinson's disease research, not to put too fine a point on it, we poison monkeys' brains. And then we try to believe that the symptoms they show are like Parkinson's disease. But the problem is, it isn't Parkinson's disease. And it now seems that the model is poor and not relevant to actual human Parkinson's disease. Now, if I can make an analogy here that I've used before, I think it's similar to making your dog sniff some pepper out of your hand. And when it gets all sneezy and snotty and its eyes are red and its eyes are running, you say, this is a model of human colds and flu. Look at the symptoms. But it isn't. It's a dog that sniffed some pepper. And I'm not shy of using this as an analogy for what's going on with injecting poison into monkey brains and thinking that's Parkinson's disease because they get tremors. So what I'll also say is I was fortunate enough to organize and conduct a roundtable and workshop with some really worldwide eminent Parkinson's researchers last year. And this is the publication that came of it. It's all beautiful, elegant, human relevant research that I'll talk about later. And there is a consensus that this is the way Parkinson's research needs to go. Again, I'll try and whiz through this. This is a really important issue. It's possibly my most read, most downloaded, most cited paper, and it's on the stress that animals experience in laboratories. And that this stress is not just a welfare issue, it's a scientific issue too. So animals in labs are stressed, unavoidably, chronically. It stands to reason given the unnatural environment for them in a laboratory and what they're going through in the experiments that they're involved in. But it's demonstrable too. So here you see stress results in an elevated heart rate, blood pressure, stress hormones. And this doesn't just affect welfare, it affects the data that you get from the animals when you do your experiments. It affects your results. So just quickly, stress is so powerful, it affects almost every, if not every physiological system, the nervous system, the immune system. In humans, it can be so powerful it causes heart attacks and strokes, high blood pressure, cancers, autoimmune diseases, and so on and so forth. So the point is this, and we accept that people are trying to mitigate stress levels for animals in laboratories, but this is the point that the paper makes. Often you can't do that. The environment is so alien, you cannot mitigate the stress. And it's even true to say that sometimes attempts at enrichment, for example, giving animals something to do in cages, something to play with, can actually cause stress rather than reduce stress. So the point is, this is a welfare issue, but it's also a scientific issue, and it's not really an issue that you can do much about. So the data you get from animals in labs often isn't really applicable to the same animals if they were living in the wild, never mind saying that those data are applicable to human beings. What I'd like to do now, again, I'm going to try and whiz through this as quickly as possible, is give some more examples of failure, this time from chimpanzees. Now, I was fortunate enough for a number of years to be involved in this campaign against chimpanzee research, to end chimpanzee research in the United States, which was the only real country in which it took place. It was a campaign of what was then NIEBS, based in Boston, now known as Rise for Animals. It's worth saying that this was a successful campaign. In spite of the chimpanzee researchers metaphorically stamping their feet and insisting that we didn't have a hope in hell of curing anything and understanding anything if we didn't use chimpanzees, the US Institute of Medicine conducted an inquiry to which we gave written and oral evidence and ultimately agreed with us and our evidence back in 2011 that chimpanzee research wasn't necessary and that they weren't going to fund it anymore. So to give you some examples, first of all, there's the failure to develop an AIDS vaccine. Chimps were central to HIV/AIDS research and vaccine testing for a long time that I've already spoken about. But as well as that, we discovered that chimpanzee research, claimed to be of vital importance by those who did it, surprise, surprise, without which we couldn't hope to tackle human diseases, was often of so little importance that it was never subsequently cited in any scientific literature. And even when it was, it didn't contribute to any reported human medical breakthrough. What did contribute to those breakthroughs, as always, was human-specific research. We also saw that, contrary to claims, chimps were almost never used in cancer research. Why? Because they rarely get cancer. And even when they do, there are important differences with respect to human cancer. In hepatitis C research, if you look at the graph on the left here, we saw that chimpanzee use had declined, while if you look at the graph on the right, the use of human-specific research methods had absolutely skyrocketed over the same time frame. Many things that had been claimed could only be studied in chimpanzees, it turned out, could be studied using human-specific methods. And we also reported why chimpanzee research wasn't reliably informing hepatitis C in humans. As with HIV/AIDS, while chimps could be infected with the virus, there were major differences in pathology, course of infection, and disease. Something very different is going on when that virus infects chimpanzees compared to when that virus infects humans. We also looked at why chimpanzees were a failure in these and other areas of research, because it was claimed that, well, chimps had to be a good model. They were nearly identical to humans, 99% genetically identical. They're not as close as was claimed by the chimpanzee researchers. It's more 94%. If you look at monkeys, it's claimed they're 93% genetically similar to us. They're not. It's actually 89%. Now, these figures might still seem quite impressive, but this is the point. Those genetic differences in the 20 or so thousand genes that humans have really matter, and they are linked to neurodegenerative diseases like Alzheimer's and Parkinson's, to the function of our immune system, and therefore our susceptibility to infectious disease and what those infectious diseases do to cancer, to HIV and other viruses and so on. But further, even when humans and chimps and monkeys share genes, those genes often behave differently. They're expressed differently, and this has huge consequences. So this slide shows many different tissues, the whole brain, parts of the brain, the liver, the kidney, the heart. And this is the point. A large percentage of the genes that those species share, thousands of genes, are expressed differently between species. And this has huge consequences that mean you can't reliably apply data from any one species to any other. Here's some more evidence. Analyses show that curiosity-driven look-see, so-called basic research hardly ever translates to human benefit. Even the most high-profile research, animal research, translates to humans very rarely indeed. Now, just last year, I published a significant amount of work that looked at media-reported animal-based breakthroughs in the UK national press in 1995. And there were 27 of them. We followed them up 20 plus years later. 26 of those 27 had not resulted in the human benefit that was claimed by the researchers. Only one of the 27 did, and even then, that was with major caveats. And that was a huge piece of work, a big paper that was published in BMJ Open Science that my co-author and I are very proud of. So I think this damning evidence alone means we should stop using animals immediately, even if there were nothing else we could do in science. But the so-called alternatives are amazing, and I'd like to now let you know a little bit about what they are. So human cells and tissues of many kinds can be grown in the lab. They have been for many, many years. It's what I used to do in my own research 25 plus years ago. And they're more human relevant because they're human cells, even than whole, complex, non-human animals. But these human cell and tissue cultures are also being improved all the time, like being grown on scaffolds for three-dimensional cell cultures for even better, closer relevance to the real living human body, an improvement that you can't really make with an animal. And many different cultures can now be grown, for example, on a little plastic or glass chip, tiny little chips with circulatory systems mimicking the blood with metabolism and so on. Now, these organ chips, as they're called, and you may well have read of them in the media, they're huge at the minute, can be connected into things called body on a chip. And they're already being widely used in research and drug development, and they're being improved. They can only get better. And things called organoids, tiny human 3D organs the size of a fly's eye or a full stop, are being grown using human cells, and they function like tiny human organs. They're already being used in many and varied areas of research instead of animals. For example, to mimic how drugs are absorbed, metabolized, and excreted by human beings, with linked chips containing three-dimensional cultures of human intestine, human liver, human kidney, perfused with circulating blood substitute. These can be seeded with cells from individual patients, reflecting an individual patient's disease, so they can reflect interhuman variability, all without the need to worry about interspecies differences and variability. These setups are already predicting drug toxicities that are not being revealed by animal tests, providing a more accurate environment for the study of human cancers, predicting one of the biggest causes of drug failures, which is toxicity to the liver, providing reliable human-specific models of Parkinson's and Alzheimer's disease, being used to identify new drugs for COVID, and much, much more. Amazing networks exist and are being set up and improved constantly to facilitate the supply of human tissues to researchers, which can replace animal tests, as well as provide valuable research material for investigating many diseases and biological processes. And we shouldn't overlook the power of computers, which can model human organs and physiological systems now, and in some cases have been shown to predict the human toxicity of chemicals much better than animal tests. And such approaches, again, are being used for many applications, including, for example, the repurposing of existing approved drugs for things like COVID and Parkinson's and other diseases. So given everything I've spoken about, why is the pace of change, which is real, we have to be encouraged by this, it's real, but why is it so slow? Why is it much slower than the evidence demands it should be? Well, there are many reasons, and these include habit, entrenched views, the hubris of some animal researchers, poor funding for human-specific methods, bureaucracy, unfair validation requirements, and so on and so forth. One encouraging point is Animal Free Research UK and many, many others are doing something about this. They're addressing all of these issues. A little bit more cause for optimism. I've already given lots of information about why we shouldn't be using animals from a scientific perspective, tempered with some real positivity about some information on what scientists could and should be doing instead, how those methods are already delivering. And here's some more to finish up. What we at Animal Free Research UK, my colleagues and I are doing to change things. On the left-hand column under activities, this is just a brief overview. The left-hand column shows what we're doing generally, though of course there are many specific projects that come under the umbrellas on the left-hand side. The middle column shows what the outcomes should be. Overall, an increasing number of scientists are committed to human-specific research, are using human-relevant methods, are staying on that human-relevant path, that there's more funding available for human-relevant research, that the number of animals being used in research is going down significantly year on year, that policymakers, industry, and the academic community are supporting animal-free research. So, for example, more high-impact scientific journals are publishing human-relevant research. More universities have animal-free medical research groups, and that we're seeing increasing discussions and actions among policymakers to replace animals with human-relevant techniques. Finally, you'll be pleased to hear the penultimate slide, I think, more cause for optimism. I've taken the liberty of posting a photograph just from a parliamentary reception that Animal Free Research UK held on Monday, at which our patron, Dame Joanna Lumley, spoke about our work and our vision and mission to shift biomedical research away from using animals and to human-focused research methods for the good of everybody, animals and humans. 25 MPs visited the reception to hear about this and about our 8 steps that we want to see implemented to achieve this. And they are, I'll quickly run through. A commission for an inquiry into the human relevance of current medical research. This is the point. There is such a huge amount of evidence out there to support this. What we need is the right people with the power to do something about it, to pick it up, to read it, to digest it, to absorb it, and to want to do something about it. Step 2, to make them ensure that UK law contains a commitment to the ultimate replacement of animal research with human methods. We want them to produce an action plan for encouraging the widespread adoption of human relevant techniques. We want them to launch support and training for scientists to do this, to get over the fear of change. We want them to shift funding away from animal research and towards human relevant research. We want them to incentivize the development of new human-specific methods and the take-up of human methods, for example, by a tax relief system, which can work. We want them to enable experts in various fields of human relevant research to be able to constructively suggest human relevant approaches to research proposals that currently use animals. And we also want them, hopefully, to appoint a Minister for Human Relevant Science who can oversee this and make sure it happens. And that, I think, is a good way to show my very final slide. I'm awfully sorry if I've overrun, but which is to show my furry and feathery friends that have lived and do live with me up here in the wilds of Northumberland, where I'm talking to you from now, and that really enrich my life and support me in the hard work that I'm doing. Thank you so much for your attention in what I know has been an onslaught of information. I do appreciate it. And I hope to be able to answer your questions and have some really constructive discussion right now. Thank you so much.
Thank you ever so much, Jarrod. I don't think you want to stop. Yeah, so I said, if anybody has questions, if they would like to type them out in the chat box, or if they're happy to be videoed, you can just raise your hand and ask a question, and I will put you on the video screen. If anybody has anything, I don't know if there's anything yet in the chat box. While we're doing that,
John's got his hand up, Brian.
Has he? Okay, yeah. John, I'm going to see if I can find you on.
Yep.
Just bear with me.
I think I'm unmuted.
Just bear with me a second.
Yep.
Yeah. Can you speak? I can't actually see you, but let me.
Okay. Yeah. Okay. I'm assuming you can hear me.
Yes, I can.
I'm partly playing devil's advocate here, but the drugs companies they want to make money. They want their product to be better than everyone else's product. Surely they wouldn't be using animal experimentation if they didn't think it was going to be of value to them. So bottom line, why do they continue with it?
We're still asking this question. There's lots of people asking this question because it always comes back to the evidence. The evidence shows that the animal tests aren't predictive. So why are you doing it? If it is of value, then use the evidence that you have at your disposal to prove your point. They don't do this. So we can only assume that it's from habit. It's from fear of change. And I've heard this said that there is a fear that let's say if the regulators jump from requiring animal data to just requiring human data and something goes wrong with a drug that's approved in that way, the whole thing will fall down. Now, the truth is it won't, because this will always happen with whatever methods you're using. They're not perfect. So you will always get something coming through that causes some problems. What you're looking for is for one approach to be better than another. And all available evidence points towards human relevant methods being more predictive than animal methods. So you use the best methods. So we have to try and understand the fear, but we have to try and overcome that fear. And actually there is starting to be a shift. The pharmaceutical companies are starting to incorporate these methods, if not necessarily in their testing and their development programs, because of their human relevance. The regulators are starting to use them themselves. They're, organ chips are in the FDA. Lung chips were being used in the FDA for research into COVID. So things are starting to happen. There is a thought that what's going to happen is there will be parallel submissions. Now, there's some resistance to that because it takes time and money, but that's the way it's going to go, just to build up the confidence for that shift to finally happen. But another point I would like to make is that the carrot for the pharmaceutical industry, that the carrots are really now there dangling in front of them. And one of them is this. There was a paper published recently that showed, I think I mentioned it in my talk, one of the biggest reasons for drug failure is drug-induced liver injury that isn't predicted well by animal tests. So you take a drug, it's absorbed, it goes straight to your liver where it's metabolized, and either the drug itself or the metabolites that the liver generates damage the liver. Now, that's not predicted well by animals, but they've shown that it's predicted superbly well by human liver on a chip approaches. And it's been shown that if the pharmaceutical industry use these techniques to look at which drugs might cause liver injury in humans, and if the regulators accept this, it can be worth $3 billion per year to the pharmaceutical industry. Now, that's the sort of thing that's really going to get them switched on. This is really significant, and I really think that this is going to be a big part of the shift. It doesn't really matter why this happens; it just matters that it does. And if it's money that takes the pharmaceutical industry to shift and give them the confidence to make that shift, then that's great. But yeah, it's a very complex issue, but it's a great question.
Thank you.
Thanks for that. And Ketan just commented, it looks like you can also bring drugs quicker to the market by skipping the animal step. I'm assuming is the case, isn't it?
Yes, and in fact, there's a couple of precedents with that. The argument that I've used in conversations in the past is that if the animal tests really were valuable in protecting humans that participate in clinical trials, you would never see them done in parallel, and you do see them done in parallel. And this has happened. Things were skipped and shifted many years ago for some HIV drugs, for some protease inhibitors. Yeah. It's there in the literature. There was pressure from patient groups to do that. And it also happened with COVID. The animal tests were done, but they were done in parallel, not before, in some instances. So it is starting to happen. But it's true that that's another issue. That's another carrot for the industry and the regulators, which is, the animal tests take time and they are expensive. They're not as expensive as clinical trials, not even close. But it's another issue. If you can do your research and your testing using human on chip technologies and organoids, it's quicker and it's cheaper, and it won't eat into your patent time, so you can make more money, hopefully, when your drug gets to market. So it's another carrot for the industry, definitely.
Yeah. There's a long, this is from Debbie. She says, the government use of public money to fund medical research, I believe, is about 2 billion annually. The Medical Research Council alone grant 122 million to animal use. Of this, 3 million is specific to use of dogs. Yet even today, when Henry Smith MP asked if enough government funding is going into NAMs, well, I'm not sure what that is.
Yes, new approach methodologies, NAMs. These are the advanced cultures I mentioned.
And George Freeman proudly stated that the National Centre's 3Rs is granted around 10 million funding, so heavily weighted to animal use.
Yeah, that's a huge issue, and one that we're trying to address with our public affairs work, and other groups are doing so too. It's still a tiny percentage of research funding goes to the development of human specific methods. In fact, most of the money that goes into it is from private venture capital and investment, and we need to see a shift. We need to see a shift in that from the government. We're not suggesting that the government magics up some more money to throw at human relevant research. What we're saying is divert it from animal research to better, more humane research methods that are going to benefit everyone, animals and humans alike. And that's something we're going to work very hard to do. And one final point, it is true, you know, the NC3Rs, a lot of their work isn't on replacement. It's not on replacing animal research. It's on reducing the numbers of animals used in research, and it's on refining that research to try and make sure that the animals don't suffer as much as they might if that refinement weren't done. The problem with that is that it doesn't address the fundamental issue, which is the human relevance of doing animal research. That's what needs to be challenged. We need to focus on human relevance. The time for the 3Rs, I and many others believe it is up. They served a purpose 40, 50, 60 years ago. The time is up for that now. We need to focus on human relevance. We could and should be doing that, and we really need to do that.
There's an organisation called Frame. I don't know if they're still in existence. And
yes,
is that a government backed? Is that?
Do you know? I'm not sure. I don't think it is, but I don't know. I might be wrong. But yeah, they traditionally have always focused on the 3Rs. You know?
Yeah. Sun wrote, do you agree that drug companies and their shareholders benefit financially from early publishing? Early publishing information about new wonder drugs that appear to work, inverted commas, in animal tests, because this often increases their share prices and attracts more financial investment. I think this ties in with what you said about your research from, was it 1995, when you look back at what was published then and what actually happened 20 years later?
It could well do. I mean, it's a very, very valid question to say, why the hype? Is it just human nature? Did we hype everything, or are there kind of, you know, are there other reasons behind the hype? Certainly in research, in the work that I did, I mean, some of the claims of the researchers, of their institutes, were outrageous. You know, this is going to be one of the world's biggest drugs in 3 years, in 5 years, whatever, and it didn't pan out. It's not necessarily the journalists' fault. They're often not scientists. They can't fact check this. But something needs to be done about this hype, because largely, actually, it's an ethical issue, because it gives false hope to the people who stand to benefit from these breakthroughs. And ultimately, the point is this, and this is an important one: when you follow animal-based breakthroughs, in inverted commas, through, a tiny proportion of them pan out. And actually, quite often, what's at the root of that breakthrough is not the animal work at all. It's the human work that was done around it.
Ketan, I think this is more of an observation, just how much does it cost, apart from suffering, to raise and look after animals for testing? This cost would be eliminated, obviously, if we're not using animal testing. I know there's a whole industry set up about, you know, the cages and everything else that for holding the animals. So yeah, it's quite expensive, and I presume the genetically modified animals are going to cost a lot more in terms of creating them, whereas compared to before, when they were using mice and rats, it would be quite cheap. So I'm assuming the price would have increased as well.
Um, there's a couple of issues there. Firstly, your kind of commercially available mice can be hugely expensive. The problem is that techniques like CRISPR, the one I mentioned, is relatively cheap and quick, and this is a real problem. Because if more scientists embrace genetic modification of animals because there's a new technique that's relatively quick and cheap, you're going to get a huge rise in the creation of GM animals, whether they're good science or not. And actually, this leads me into something else that was in the news recently. It might have been the MRC, but I can't remember who it was. Finally, this often happens. A group of scientists or a funder will agree with something we've been saying all along, but not take the right action. So, for example, there was a group of scientists, I think it was the MRC, said, quite often GM mice aren't really relevant to humans, so we need to change this. We need to do something instead. So instead of focusing on human research methods and human biology, they've decided to make a load of GM rats instead. And unfortunately, and I'm going to be candid here, I think sometimes science can be so clever in some ways, but also so just downright stupid in some ways. And my opinion is that shift from GM mice to GM rats is just stupid. It's not backed by evidence. It's just crazy. And there are times I try to convey some causes for optimism that are genuine in my talk as well. But sometimes you get things like this happen where you just think, what do we have to do? What has to happen for this shift to take place?
Yeah. Thank you for that. Any other questions? If you put your hand up, we've got about a couple of minutes. If you have, I'll take a. Well, there's one here. Actually, there's two. Ketan says, would it be best to have a focused approach to eliminate animal-specific diseases, like on a one by one? In other words, try and tackle cancer research without animals and then moving on to something else rather than just a general
I think that's
avoiding animal experiments altogether.
That's a really nice point, and that's something that we're looking at, to be perfectly frank. And there are two reasons for this. One is the direct impact that would have on the disease itself that you focus on, which I think would be hugely positive. The second thing is the indirect effects that would have, because if there was an area of research like that, that was really taking off and in which breakthroughs are really happening, other scientists in other areas would kind of peer in and say, what are you doing? How is this just taken off? And they would see how. So it would kind of osmose, if you like, across into other areas of research. And by the way, I think this is going to happen. I think I see scientists' heads being turned now into these new technologies, and they're amazed by them, and they want to be part of them, particularly young scientists. And I think very, very soon we're going to hit a critical mass where this just snowballs and takes off, and animal use will relatively fall by the wayside as a result. I think people like us will always wish it would happen more quickly, but it just needs to start to happen significantly. And I think that's going to happen.
Thank you. Margaret, let me just, if you would unmute, let me just spotlight.
Yes, well, first I want to thank you for your great work. It's just fantastic, all that you've done. I'm standing here because my cat has taken my chair. But I had a very close friend, a dermatologist, he died a couple of years ago, but he told me a story of his time in medical school that has always stayed with me. He was someone who loved animals, had a great heart for animals. And he said that when he was in medical school, he had a professor who forced him to do a terrible, terrible thing to, I believe it was a cat. And he tried to resist doing this to the cat. He knew that there was no good reason for it, but the professor was holding his degree over his head. If you don't do this, you won't get your degree. I took that as almost a macho type culture that existed. I don't know if it's still like that, where you have to prove that you're strong, that you can do anything required in the name of medicine. And It, I just, that story lingered with me because I wondered if that's the culture that is one
I,
of the barriers that you describe.
I think that I've spoken about this before. There certainly is. This is published. This is a phenomenon of it's called it's both individual and institutional lock-in. You just get locked into a way of doing something, and you get kind of caught up in it institutionally in your research group. Not everybody, of course, but some people do. So you do get caught up in that culture. I think one important point I'd like to make that this brings in, this brought to my mind is a culture in scientific publishing. And again, this is something that we, with some other groups collaboratively, are starting to address now. We've actually had a meeting about it with some journal editors, and we're hoping to publish it later this year. And it's on publishing bias. So, for example, if you publish some beautiful research that you've done with a human focus, let's say using organoids or human organ on a chip technologies, you will probably get a reviewer who says, well, this is all well and good, but it's in cell culture. Now you need to go and prove it in an animal, and we're not going to publish it until we get the animal data. And this happens, and you also get it with grant reviews as well. If you're applying for some grant funding for your research, people will say, can you factor some animals into that for proof of concept? So we're trying to change this because this is the wrong view. This is the wrong culture. This needs to not happen, and we need whether it's journal editors or grant reviewers or whatever, we need people to take the science on face value, and that does not have to involve the use of animals. So there is a cultural thing there, but we're trying to change that, and we're confident we can.
Thank you. I've got a couple of let me spotlight myself. Yeah, there's a couple more questions, and then we'll wind up for this evening. So Atan asked, what type of animal testing is currently prohibited in the UK? Assuming, I think there are some, particularly primates, I think, or some that may be different for warfare experiments, I think. And secondly, worldwide, which pharmaceutical companies are the worst offenders? You happen to know, I don't know.
Well, I can't answer the second question. I really don't know. I mean, there are some really encouraging pharmaceutical companies. Like some people here might be aware of a company called Vanda in the US that stood up to the FDA when they asked for some dog tests, and they went back to the FDA and said, your request for these dog tests is not scientifically valid. We don't need to do these tests. They will not tell us anything. And the FDA told them to just do them. So they took the FDA to court, and of course they lost, as you might expect. But this was a real groundbreaking moment because it was a pharmaceutical company standing up to the FDA, requiring them to do an animal test that they knew was not scientifically valid. So I think we might see more of that. Just to go back to the kind of research that doesn't go on in the UK. Actually, there's an interesting anecdote about this. So I mentioned that I was very fortunate to be part of the chimpanzee campaign in the US that was ultimately successful, and the NIH stopped funding chimpanzee research. When the EU raised a directive to ban great ape research in Europe, there was a lobby in the UK to try and get a derogation in UK legislation, which was if, for example, if some eminent scientists decided that they needed in an emergency to do some research on chimpanzees, on great apes, then they should be allowed to do it, this derogation clause. And we managed to get that clause taken out of UK legislation, largely using the evidence that we managed to, that underpinned our victory at the Institute of Medicine in the US. So thankfully, great ape research, it didn't happen anyway. We have to say that. So we kind of got something stopped that wasn't happening anyway, but we stopped the possibility of it ever happening. So that doesn't go on in the UK. But I think it's interesting times. The UK is now starting, maybe because of Brexit, who knows, but it's starting to want to cement itself or even become this global scientific superpower. And one of the arguments we're using is to say you can do that, But if you want to do that, you have to go down this route because this is the future of science. You don't go down the route, for example, that China is going down at the minute, which is come and do your monkey and your GM monkey research here, because you won't get as much trouble as you might in the UK or Europe. You can do it largely unhindered. So I think the point is, we have an opportunity that we need to take. It just remains to be seen whether we take it or whether we just continue as we were, which I think would be disastrous for everybody.
Thanks, Jarrod. One final question, actually is part of Son's question, which I think maybe, I'm not sure if it is within your remit or not, and that's the use of genetically modified animals for the purposes of transplantation. And I was in the news not that long ago about somebody who had a part of a pig's organ. I can't remember which organ it was.
It was a heart.
And then he subsequently died, I think. So the question is, is that something that we can do something about? Is it, I suppose it's a question of, there are a lot more pigs' hearts available than human hearts that are available for transplantation, but is it going to be of use, or is it just going to be going down a dead end and then they realize they've killed all these animals and the humans at the same time?
This is actually part of the paper I mentioned in BMJ Open Science on media hyped animal-based breakthroughs. Xenotransplantation, as it's called, is one of the topics. So you can find that paper and have a look yourself. In a nutshell, and I looked at this years ago actually for another reason. This has been attempted for decades, and it's just never worked. And there are very complex but very good reasons for that. They obviously involve rejection of the organ. Now, researchers initially thought they got around that, but then there was another mechanism of rejection came into play, and they thought they got around that. There was something else came into play. There are endogenous pig viruses that can cause issues. There are issues of the level of immunosuppression you need to tolerate an organ from another species. There are scientific issues with the degree of genetic modification that you have to do to stop your immune system recognizing all these foreign things and rejecting the organ. I think I would say I would be immensely surprised if this ever worked. I just think it's a crazy thing to do. Now, that's not to say I don't sympathize with the aim. There is a shortage of organs for transplant. But I don't think that this is going to work. I think it's too difficult. I think if it ever does, there'll be huge caveats to it. And I think, along with many others, that there are better approaches to doing this. And they include societal things like presumed consent or opt-out, and also scientific solutions like bioprinting, 3D printing of tissues and even eventually organs. That's where the money should go, not on genetically engineering pigs in the hope that you are going to get around very complex rejection mechanisms and huge amounts of suffering, by the way, for the animals involved, not just the pigs, but the non-human primates that are used as the organ recipients in the research.
Thank you ever so much. I'm going to leave it at that. I appreciate there were a few other questions. We haven't got time to cover everything. Can I ask if people be kind enough just to unmute for a moment? And I'd just like to say a big thanks to Jarrod for presenting to us. If we could give him a round of applause, if that's...
Thank you.
Very much.
Thank you so much. Thank you for sticking with it and the onslaught of 42 slides or whatever it was, I think, and for the very intelligent, good questions at the end. I really appreciate that. So thank you.
Just say, if you'd like to support Animal Free Research UK, if you're going to visit their website, I will send out a link when we do our next mail out. So if you want to help support them financially, please do so.
Sorry, just a final thing. I did mean to say on my last slide there to check out our website. There's all sorts of information on there. My email address is on the last slide. And also, if you want to have a look at any of the work that I presented, the scientific papers are at a website called ResearchGate. And if you search my name, you'll find my page and you can access all of the work. So thank you.
Thank you, Jarrod. I'm just going to